Good morning, everybody. Thank you very much for joining this webinar that's been organized by the ATS on IPOG, or Interventional Pulmonary Outcomes Group. It's a pleasure to have you. I'll be your host today. We have fantastic speakers. And really the point of this is to give you an introduction on what it is we've been trying to do with IPOG, what IPOG is as a secret society trying to take over the world primarily, but also as we hope, an increasingly inclusive group that will help foster research in interventional pulmonology. As a brief background, IPOG really stemmed from an effort to coordinate efforts that were isolated between different research groups that were primarily interested in bronchoscopy, realizing that we're usually more than the sum of our parts, and that federating and integrating efforts is helpful for research. I think we've reached a maturity point where we're now very happy and enthusiastic about including a number of other members and institutions that have the same interest in research. And so really the purpose of this today is to introduce you to some of the work we've done within IPOG, and mainly to try to motivate you to join and participate and collaborate with IPOG, introduce your ideas to the group, and try to leverage the resources available within IPOG to get this project to completion. That's really the point. So without further ado, I'm going to introduce the first speaker. We have four fantastic speakers today. Jason Akulian will go first. We'll have Jeff Thiboutot, Rob Lentz, and Diana Yu following. Jason Akulian is an associate professor at UNC's director of the Interventional Pulmonary program there. He's got a specific interest in pleural diseases, which is obviously a very hot topic and one that really lends itself well to a lot of research. So Jason, take it away.
All right. Jeff, I think you're firing up the slides. Is that right? Thank you, Fabien, for the very nice introduction. And I'm going to frame this as I'm going to talk about pleural disease, but I'm going to focus on infectious pleural disease just because the field is so large and so long-standing that if I were to try to dip my toe into malignant pleural disease, we probably would run out of time a long time ago. So with that, we can advance slides. These are my financial relationships. So like I said, I mean, this goes back a long time, right? So way back into the mists of antiquity. If we look back as far as basically the first century BC, the idea of treating pleural disease first is brought to us by Hippocrates of Cos, right? So the same gentleman whose oath we take to do no harm, even though it doesn't exactly say that, was also really interested in taking care of those Greeks who developed empyema, and noted that if you don't treat the empyema, then will actually rupture. So he actually described empyema necessitans. He also advocated the use of olive oil and wine-soaked rags to be shoved into the pleural space and ultimately slowly retracted over time, thereby obliterating the space. So, as you know below, pretty much everyone got pleural disease back then, and they died quite quickly. But, I think of maybe Hippocrates as being the founding member of IPOG, as he tried to pull together the Greek Peloponnesus to figure out how to treat this. We'll go to the next. So time goes on from there. There's a lot of death and war, and I'm sure a ton of pleural disease as people had penetrating chest wounds. And it wasn't until about the 15th century when Perret actually sort of recapitulates what Hippocrates had looked at a significant period before, where patients who develop pleural disease would develop fistulae and then ultimately would succumb to their complications. It wasn't for another 200 years before anyone actually started to write more about this. And there was actually some pioneer work in mouth pipetting the pleural space on the battlefield. So those little drummer boys that you'd see depicted in these old pictures, their secondary job after they were done drumming and the battle was over would actually be to clean up wounds and to try to help people on the battlefield. And he pioneered they did a lot of mouth pipetting. He ultimately introduced a piston syringe, so they didn't have to do that as much anymore, which was, I'm sure, quite welcome. about 100 years later, there were a group of physicians who sort of individually and in concert in some cases actually started to develop the idea of a closed water seal drainage, irrigation of the pleural space, suction, and sterilization, right? So they began to realize or understand germ theory, and that they needed to address the underlying cause of the disease process to improve mortality. 100 years from there, we have the flu pandemic that occurs. Along with the flu pandemic comes the strep pneumo pandemic. So strep pneumonia, also known as the old man's friend, was one of those organisms that would come on the heels of the H1N1 influenza pandemic, and ultimately there was sort of this epidemic of empyema, which prompted the creation of an Army medical commission to try to treat empyema more effectively. So we'll go to the next slide. So that commission really, popularized the idea of surgical intervention and open pleural space and to improve drainage. And things sort of stayed that way for a long time, for about 70 years, maybe a little even more. We can talk more about where we went with that. And in 1972, there was an intrepid young fellow from the Johns Hopkins who, as he were to tell it, was sort of asked to present something when there was a famous speaker coming, and so he spent all night, he said, looking at a data set and using a slide rule, because people weren't using calculators back then, to figure out if they could better characterize the source of pleural fluid and the accumulation of pleural fluid. And so, as you all are aware, the Lentz criteria was born, and that really was or became the landmark study that was able to define the etiology or the underlying driver of pleural effusions. So we'll go to the next slide. About 20 plus years later, we began to see prospective trials in pleural disease with the MIS-1 trial, where streptokinase was injected into the pleural space. It was shown ultimately to be ineffective, but the data that we got out of this trial that was interesting, or probably then used, was that large chest tubes hurt. And so we recognized that smaller bore chest tubes were just as effective in draining the infected pleural space, but that streptokinase itself didn't really work. We'll go to the next slide. This was followed up by the MIS-2 trial, which I think is probably the more famous of the two trials. Lead author was Dr. Rahman out of Oxford, which is a 4-armed multi-center trial within the NHS, where they leveraged this sort of broad coalition of research hospitals within England to perform this 4-arm study where they randomized patients to TPA plus Dornase after they'd kind of figured out that maybe there's a biofilm issue, and that was maybe why streptokinase didn't work so well. But they showed that TPA and Dornase alone were not terribly effective. DNase by itself maybe even had a harm signal, but that the combination of the two improved X-ray findings, and that the combination may actually reduce hospital stay, sepsis, and/or the need for surgery. This was followed by a number of retrospective trials that sort of, again, recapitulated the findings of the MIS-2 trial with high degrees of success. But maybe the story is not always told with single center retrospective trials. And so, we started to think about that and try to understand if there was maybe some additional measures that needed of this approach that needed to be performed. So go to the next. Around the same time, we had some clinical prediction models that were beginning to develop. And again, this was all coming out of the group in Oxford, and it was, they've really been a model in many ways in terms of how these sort of collaborative research projects can be developed and then grown. The rapid model was developed in 2014, sorry. And it was derived from 2 large data sets. Was able to risk stratify patients presenting with pleural infection into low, medium, and high risk strata. Didn't really give any guidance about what to do with those folks, but it did give folks a sense, or give clinicians a sense as to where they may risk stratify patients based on their 90-day mortality. This was then prospectively validated, and again, seemed to bear out. We know that anytime we go from retrospective looks and development of prediction risk models into a prospective evaluation. The curves begin to move together, but this remained a positive trial in that it was able to show that you were able to still risk stratify these patients accordingly. Now, of these patients in the pilot study and in the rapid scoring, there really was a small percentage of them who received intrapleural enzyme therapy. And so this begins to make one ask the question, what's the effect of enzyme therapy or TPA and Dornase on the outcomes that the rapid and pilot study showed. So next. And one more. So as one of the sort of, I'd say, first big attempts to put our foot in the water from an IPOC perspective in pleural disease, we put together the Retrolysis Project, which was 24 centers split between the US and the UK. And it was a retrospective look, trying to really get a real world sense of the practice pattern as well as outcomes associated with the use of TPA and Dornase in patients with pleural infection. So these were the inclusion criteria as well as the exclusion criteria. We looked at patients essentially from the beginning of the MIST-2 trials publication until May of 2019. There was a viral pandemic that happened not long after that, so we closed enrollment and then had a large data set to analyze. Next. Our first publication that came out of that looked at over 1,800 patients. They're able to quantify the risk of bleeding for this therapy, right? So that was actually one of the pieces of feedback that we got when we sort of did a back of the napkin survey of pulmonologists who were using or not using the strategy of TPA and Dornase, was why they were or were not using it. And the sort of overarching response as to why people were not using it was this question of bleeding. And this number two was sort of the off-label use and whether one's hospital would allow them to use it. And so we're able to actually quantify the overall risk of bleeding as well as those factors that were associated with increased risks of bleeding. And I think this sort of helps reassure folks that that there isn't a sky-high risk and this isn't something that is outside of the bounds of practice. Continue. Our next phase of looking at this data set is really then to try to understand whether or not we can predict failure and mortality. We have a current manuscript that's under review from the Blue Journal, looking at 732 of the 1,800 patients enrolled. Now, we lost about half our enrollment because of incomplete data, and we really wanted to develop a risk stratification score based on complete data sets. The primary endpoint is 90-day IAT failure, defined as 90-day mortality and or surgery. So either knife to skin and or death at 90 days. And ultimately, we have some secondary endpoints that we were able to show good prediction scoring on. Hopefully, we'll see that actually in print very soon. So I'm going to sort of wrap this up and just sort of thinking about where we've come from antiquity to now real world. I think IPOG is a really great opportunity to continue to define our disease space, right? So I think, retrospective single-center trials, they have their place, but I think we're now in a space where we need to be looking at this from a multi-center and as preferably prospective approach. We're currently standing up something called the Only Once trial, which is looking to see if we can dose TPA and Dornase once daily versus twice daily. This is currently working its way through our IRB, and once that works its way through our IRB, we'll be looking for centers who are interested in enrolling patients. This is a great space for young or even established investigators who want to get into the field and want to work closely with people who will and are able to enroll and stick to a protocol. And there's other opportunities here for branching into basic and translational research as we develop biorepositories and more capacity to ask questions. So with that, I'll thank everybody for having me.
Thank you, Jason. I am monitoring the chat, so if you have any comments or questions, feel free to enter this in the chat. If you raise your hand, I may or may not see it, so I may depend on Samantha to alert me of that. I think these innovations or efforts in research at IPOG are really driven by the clinical need. So we're dealing with diseases with a very high burden of morbidity and mortality, like pleural diseases and lung cancer. The other driver is the gap in technology, and in that we are trying to partner with industry either during the R&D development or afterwards when the device has been cleared by the FDA, is already in use, and try then to generate meaningful data on clinical outcomes. So the first of these two remaining talks here will be specifically about the partnership, which is really kind of both pre and post commercialization. So Jeff has been instrumental in driving the cryobiopsy domain of research, very interested in diagnostics and therapeutics involving cryotherapies in general. We're very happy to have him speak next. So Jeff, go ahead and take it away.
Yeah, thanks so much, Fabien. Yeah, so what I'm going to talk about today is really a story of partnership more than anything, and on multiple fronts. Maybe most importantly, this is a big academic and industry partnership where we saw a need from an academic side, and industry saw an opportunity to modify a device to make it more widespread, and we really ran with it. And the other way we were partnering is really when we're starting these bronchoscopic large-scale trials, we're now able to loop in multiple different centers and other places that maybe traditionally have not been doing a lot of research. And because we now have the ability to design these larger trials that are going to require higher amounts of patients, that opens up opportunities for everybody to really get involved in these pragmatic clinical trials. So I'm going to go over some of the publications that have come out through the IPOG group as they particularly relate to cryobiopsy. All the data that I am going to talk about today are all multi-center efforts, all under our IPOG umbrella. So with that, just trying to frame what we're getting at and why we're doing that. Remember who we serve here. We are trying to get biopsies that are going to help our patients, that are going to help the pathologists make the most appropriate diagnosis, and help our oncologists and ILD doctors, lung transplant doctors, really secure the diagnosis and get our patients onto treatment. So how does cryobiopsy come into play here? The whole mantra behind this, or thought process behind this, is that we are able to get larger biopsy specimens. There's less artifact, there's less hemorrhage that are there. Initially, cryobiopsy was really thought of as an intermediary to surgical lung biopsy for patients with interstitial lung disease. But one of the first things that we started to recognize is that there's going to be a whole lot of indications, in fact, maybe even a lot more indications outside of interstitial lung disease. And one of those that we started to identify was in the lung transplant population. So this is with the traditional cryobiopsies using the 1.9 or the 2.4 probe with en-bloc poles. This was an early study that we did here at Hopkins. This actually predates myself, to tell you the truth. Just showing that is the safety and feasibility of doing this in that population. We showed that there was really low bleeding rates. It was a safe procedure. This study actually had patients undergo cryobiopsy and forceps biopsy at the same time. We did actually pick up on more cases of rejection in the cryoprobe, in the cryobiopsy arm itself. So lung transplant, moving on to a different population, still outside of ILD. This is more of the the nodule masses, lung cancer side of things. So this was another IPOG study that came out with our European and Asian colleagues, where we put together over 1,000 cases that had been performed. This is all for targeting peripheral pulmonary lesions, so nodules or masses. These were a cohort rich in cancer diagnoses. And the primary outcome of this study is really to look at safety. Could we go after masses and biopsy them safely? Again, this is also with the larger 2.4 probe or the 1.9 probe that does require those en-bloc poles. What it showed is that we were able to get fairly high diagnostic yields and relatively low complication rates, low bleeding rates, and pneumothorax rates that were on the order of like 5–6%. With that, we've also published a few studies and some of the guidelines that have actually come out through the ACCP for cryobiopsy in the interstitial lung disease population. This is a study that was performed largely out of Duke, Northwestern, and at Johns Hopkins. And this is a similar study that was set up to the cold ice trial. This was a chill study where patients in one setting underwent sequential forceps biopsies, cryobiopsies and surgical lung biopsies. And after all of this data from each one of these different modalities of biopsies was presented to a multidisciplinary group, there was decent concordance between each one of them. So it shows that there's definitely a signal there that it can help in the ILD population. But what's the big problem with this procedure? I'm sure everybody's seen this slide or a version of this slide in the past. It's really that cryobiopsy with the traditional en-bloc pulls is a bit of a clunky procedure, to tell you the truth. You do have to be fairly specialized with your skill sets. The safest way is likely to do this through a rigid bronchoscopy, if not using a large bore ET tube. You need to have a bronchial blocker in place. And then you really need to be savvy about once your scope and your specimen is all pulled out of the airway, that you got to get back in there quickly to make sure that there's no complications. So pulling together all of the data for this is mainly for the ILD population. What it showed was, yes, our yields are higher with cryobiopsy, but our pneumothorax rates are about twice as high. Our bleeding rates are about 5 times as high. So this really led to the innovation and the change, and really the partnership that happened between us and our industry partners about how can we improve on this device? How do we change the technique? Do we need to redesign this product? And then even after we design it, how are we going to move it forward so it can actually be commercialized and get out to the general public? We need to show up as a feasibility. We need to demonstrate safety. Finally, which is the phase that we're in now, is proving efficacy so we can expand this to different populations. So what we designed was actually a miniaturized cryoprobe that would eliminate the need for the en-bloc pull. So now the cryoprobe could be used in a very similar fashion to how forceps is used. The scope can remain in the airway, and the specimens can be pulled out through the working channel of the scope itself. This was actually done in collaboration with some of the Johns Hopkins biomedical engineering students here, where they actually designed and patented the original device and built a prototype of it, which eventually went on and was passed off to our industry partners for further modifications and to scale it up. Once the prototype was built, one of the first things that we did was we actually tested out in a preclinical model. So this was in a porcine model where we did the 1.1 millimeter cryoprobes, which is going just right through the working channel. We also did that, and then we did the traditional en-bloc pulls with the larger, and also did standard forceps biopsies. What it showed was that compared to forceps, all of the cryobiopsy specimens were larger, were of higher quality, and there was really no significant difference between the miniaturized cryoprobe and the larger traditional one that requires an en-bloc pull. So this was enough feasibility signal that we could move on then to our in-human trials. So after the prototype was built, again, the patent and technology went over to our industry partners. They worked it, they redesigned it, they scaled it up. It went through the FDA 510(k) clearance pathway. And then we started our Frostbite 1 trial. So this was the first in-human trial. This was a trial done at 2 centers. We did 50 patients. We included pretty much everybody who had any indication for undergoing a forceps biopsy. So these were patients with diffuse lung disease. These were lung transplant patients and patients that we were also biopsying nodules or masses. We weren't looking for efficacy here. We were purely looking to see what is the side effect profile. A lot of this came because the reason we were looking at the side effect profile was all of the fears and some of the, mishaps that were happening in the past with traditional en-bloc cryobiopsy. So what did it actually show after we did our 50 cases? Our primary outcome was pneumothorax rate and bleeding rates, and they were both exceedingly low, to tell you the truth. We had 2 pneumothoraces out of the 50 cases. Neither of them actually required admission or chest tube placement. They were just monitored and sent home. And what was actually most surprising was there was no significant bleeding, not a single case of it. Yes, there's always going to be a little bit of bleeding anytime you take any biopsy that was just cleared with suctioning. But nothing went beyond that. There was no need for ever a bronchial blocker to be placed. There were no adverse outcomes. Nobody was hospitalized due to the results of due to the procedure that they underwent. And with this now strong safety signal, we moved on to the larger study, our Frostbite 2 trial. So Frostbite 2 is a randomized controlled trial, randomizing 1-to-1 of forceps versus the 1.1 millimeter cryoprobe. This has been done and enrolled at 10 different sites. We enrolled 500 patients. Enrollment ended about maybe 3 months ago or so. We're in the data analysis phase right now. Again, very broad inclusion criteria here. So just highlighting the pragmatic nature of this study, that this really was all comers for bronchoscopy. So we stratified them into diffuse lung diseases, patients undergoing transplant surveillance or for-cause biopsies, and then nodules and masses. The majority of the nodules masses were all done via robotic bronchoscopy. Our primary outcome is a centrally adjudicated outcome of diagnostic yield. So just going over some of the mechanics and the highlights of this study, and this is the process that we're undergoing right now. Enrollment is complete right now, and we are undergoing central pathology adjudication. So we are no longer relying on all of the individual reads from all of the individual sites. We are getting all of the slides digitized and sent to a central pathological read. So they're all being assessed for diagnostic yields to make the data more uniform and transparent. And another big part of this study, which is what we're all trying to include in our IPOG studies going forward, is really safety oversight with the DSMB. So we had regular DSMB meetings all along the course of this. There were really no significant events that happened. So hopefully the data for this will be out in the next couple of weeks to months. So keep an eye out. Moving forward, this is a trial that is currently enrolling, and one that we are looking for additional sites. So if anybody is interested, please reach out. This is one of those trials that is also very pragmatic, that if you have a 1.1 millimeter cryoprobe and you are doing EBIS, this may be a trial that may be of interest to you. So this is a trial of intranodal cryobiopsy. So we're randomizing 1.1 millimeter cryoprobe to your standard 21 gauge needle. The primary outcome here is not diagnostic yield, but it's efficiency for NGS. We're aiming for 250 patients total. We are currently only enrolling at enrolling at one site, and there's 2 or 3 others that will be open and enrolling probably in the coming weeks. But we would like to get another at least probably 10 sites on board if that is possible. So again, reach out if there is interest. So overall, to wrap all of this up, what are we trying to do? We're aiming for a bit of a paradigm change here of how you think about cryobiopsy. Traditionally, cryobiopsy was thought as an intermediary to surgical lung biopsy for the ILD population, but we're really trying to change this. And the way that we're thinking of cryobiopsy now is anytime that you think that you're going to be needing or using forceps, a 1.1 millimeter cryoprobe is a viable, safe alternative to that. Efficacy data will be pending, so keep an eye out. Thank you.
Thank you, Jeff. I don't see any questions in the chat, so I'll keep talking, and I see that Lonny joined as well. So welcome, Lonny, and we'll have you say a few words as well. To me, the cryobiopsy story is great because it illustrates the best and the worst of what we do as interventional pulmonologists. So started out with these giant cryobiopsies where you pull everything, you get a big chunk of lung, and major bleeding happen, and nobody reports it for 2 or 3 years until you get the first publication with catastrophic complication, and then everybody takes a pause, starts guidelines and studying the thing correctly. The best of the story is, of course, what Jeff has done in collaboration with a number of us, looking at identifying the problem, identifying solutions, potential solutions to the problem, developing it, and starting studies in animals and in feasibility, safety, finally a pivotal trial that may or may not change practice, but at least we'll get the definitive answer. I think that's great. I'll just give another kind of brief background on IPOG because a lot of people ask, what's IPOG? How do I get involved? Well, for years, I've heard of IPOG for many years also. And for many years, it was, hey, we just finished a study. We should just stamp it IPOG. And that was pretty much the extent of it. It's only been about a year or two that IPOG has been more formally established with processes in place and infrastructure to run clinical trials. And this is why, we're having this kind of events, because as Jeff pointed out, we cannot do these studies without the right stakeholders participating, particularly people who do a lot of clinical stuff and not a lot of research, because this is where clinical care happens. And so these studies are intentionally designed to be as pragmatic as possible. That means that they should be doable with minimal or no funding, but also much less efforts and resources that conventional explanatory type trials would take. And so in that line of thought, so we'll introduce Rob Lentz. Rob is Associate Professor of Medicine here at Vanderbilt. Has been instrumental in running and collaborating with a number of bronchoscopy trials. So I'll talk about this kind of third arm of IPOG right now, which is the bronchoscopy research. Go ahead, Rob.
Thanks, Fabien, and thanks everyone for taking a little time out of your morning. All right, let's go next. Jeff, are you driving? Sweet. We can go next. All right, I see I've got some consulting from Intuitive. All right, so I'm going to kind of give, as best as I can, a kind of a 10,000-foot view of where we've been with research in advanced bronchoscopy, kind of where we are now, and then the prospects for the future through the lens of IPOG. Next. All right, so I'm not going to go through all these, I promise. But I was trying to figure out how to get on a single slide here a snapshot of kind of where we've been, and this was about the best I could come up with. So this is using total. This is studies published in the last 25 years with bronchoscopy in the title or topic, ranked by number of citations as a surrogate for things we feel are valuable as far as data in our field. Probably not a perfect surrogate, but there we go. And I'm not going to go through the details of any of these. I'm going to focus here on the methodology here and how robust is our data when we're considering the things that we value or the things we've been able to produce, right? So, and I'm not going to denigrate any of the researchers here or the studies that are done. All research, no matter what the design is a big lift. I think this speaks more to what kind of systems have been in place to support research, and I'm going to argue here that in the last 25 years at least, we haven't had great systems in place to develop the most robust study designs and the most robust data. So on this list, there are a few RCTs, two of which are valve related. Valves are a very, very narrow slice that has done very well. I think is almost more of a COPD story than a bronchoscopy story. Those two valve studies inform 40 or 50 of my 2,000 cases a year performed in my institution. What about the other 1,950? So we've got one other RCT that's a single center about distraction during MAC bronchoscopy. Let's go next. Got one meta-analysis, as I'm sure everybody here has read, on yield for navigation bronchoscopy, including mostly single-center retrospective studies. So poor quality, highly biased underlying data in this meta, all using different outcome definitions. So difficult crowd in which to do a meta that will inform a lot. Then we have a couple big prospective observational trials, Navigate and Acquire, almost always cited together as examples of how discrepant our data is. These were both on the same-ish topic of yield for navigation bronchoscopy, produce very widely discrepant results. So overall, I'd say that our study designs, what we've been able to muster with the systems we have over the last 25 years is perhaps less than it could be. Let's go next. This is, sorry, back one. Okay, this is citations per year, so just a little bit of a different look at this. I'm not going to go through all this again. We're getting a little bit more contemporary data here, but still largely, larger prospective observationals, but we're still lacking in some rigor. Okay, next. All right, just as a frame of reference, and I don't mean to argue that this is where we should be, certainly not right now, but to give you a sense of another interventional field, interventional structural cardiology, this is their top 10. where even their registries are hundreds of thousands of patients. So that's just to kind of give an idea of maybe where we can aspire to one day. All right, so that's where we've been. Where are we now? So I think this is best exemplified through the story of robotic assisted bronchoscopy. So the robots were first cleared, the first one, Monarch, in early 2018. Like nearly everything we use was cleared by the 510(k) pathway with the FDA. And this is a pathway that is a little bit less intense in regulation than a completely new device. Where you can market your device, you can sell it if you can suggest to the FDA that it is substantially equivalent to something that's already out there on the market. And in this pathway, there's no requirement for a robust demonstration of effectiveness of your device. So this is, I think, a well-intended pathway, right? So if you change the knob on your robot from red to blue, and nothing else changes, do you really need to go through a big regulatory lift for that? No. Where exactly the line is by which you should undergo some efficacy studies before something comes to market, I think is a question. But be it as it may, this is what we have. And this is very much a double-edged sword. We get a lot of new and advancing technology faster to market because of this, which I think many would argue is a great thing. The problem is we don't really have any data on what these devices can do when they come to market or land in our bronch suites. And because of this setup, there's little impetus for our industry partners marketing they spend more on marketing than they do on studies because they don't have to do the studies, right? So we're in a situation, I think, very clearly now where the pace of what is coming to market has very, very much outstripped our ability to generate robust data on what it is these devices are able to do. This is just an example of this kind of 510(k) predicate device web. So one afternoon while procrastinating writing Veritas, I decided to go back for some other talk and look at how ION was approved via the 510(k) pathway. So you can go on FDA's website. Well, you used to be able to go on FDA's website, and you could go back and it would look at their pre-market approvals, and it would tell you what the predicate device was that they were referencing, and then you would go to that device and you would see what predicate device it was referencing when it was 510(k) cleared, and on and on and on. And so, at least online, you can trace all of these devices listed on this web were 510(k) cleared based on predicate devices before them, all the way back to 1980 with early Olympus fiber optic scopes. And the trail there runs cold online. I'm sure there's paper somewhere in the basement of the FDA that goes all the way back, probably to the original bronchoscope. So you can see the quality of the image produced by essentially what is the predicate device for the ION, but this is how the system currently works. All right, so let's look at robotic research. So this is all post-market, right? There is no requirement for these studies to be done beforehand to demonstrate efficacy. So this is might not be exhaustive, but pretty close to what's been published. It's over 50 patients on robotic diagnostic yields. You can see that there's a very wide variety of outcome measures between these studies, which makes it a little bit hard to compare one to the other. Next slide or next. Okay. So all of these that I'm now kind of shading out here are non-comparative studies. Informative for sure, but with significant inherent biases, which limit the conclusions one can reach, particularly in a diagnostic yield type situation, right? There's a ton of selection bias. There's a lot of publication bias. There's a lot of unrecognized confounding in these trials. And again, it's hard to compare one to the other, and they're mostly single center because the outcome definitions are different. And we know from modeling and now real world data sets, if you apply different diagnostic yield or accuracy constructions to the same data set, you can vary the results you get by 20 to 30% alone. While this data is certainly important and helpful, it is not as robust as I think we need to inform care of our patients. There are a couple comparative cohort trials, one retrospective, one single center, get us somewhere, but maybe not where we need to be. The first of these was 21 months after the Monarch market release. So we went 21 months before we had a series of more than 50 patients showing us what these devices could do. The first randomized trial related to robotic bronchoscopy was not registered until 5 years after market release. This is Reliant, and it will not be publicly made available until ATS in May. So 7 years after market release, it took us to generate randomized data on the hottest new thing that we could possibly imagine in our field. So if we want to keep up with the technology that we're getting in our current environment, I think we need large collaborative efforts, and that's what IPOG is about. We need folks who are interested in pushing things along, and that doesn't mean that everyone on this call needs to go out and write 5 protocols and try to shovel it through your IRBs. But becoming part of a collaborative group and being interested in stuff that comes along, not even all of it, is certainly what we need, I think. I think that we need more innovative trial designs that can get trials done faster, so pragmatic or embedded trials, as opposed to traditional, explanatory trials. So Reliant, for example, was a cluster randomized trial. We randomized OR days instead of patients. We enrolled 440 patients in a year. Veritas, I'll talk about here in a minute, was a more traditional trial. 7 sites took 3 years for half as many patients. We need collaboration with industry. They're driving the innovation in our field, and so that's certainly important. And then a system that helps generate ideas, foster protocols, etc., from the very conception to their, pushing through IRBs and then disseminating them across the network, I think is ultimately what we're going to need here. Next. All right, this is just a look at some IPOG involved projects, multi-center work, some first in human or some kind of novel devices. Again, we would like to be a place where industry can come up with something and say, hey, you have a research network, a pre-made research network we'd like to study. We'd like to get this device in your hands to get it out and see what it can do. And then at the bottom here are kind of the more robust, as Fabien said, the last couple of years, everything's kind of really come together, and we're starting to incubate some very robust trials, and the 2 frostbite trials Jeff already talked about. Cryostasis is absent on that list on all the lists I've shown you so far are any trials of therapeutic bronchoscopy, excluding the valves, but traditional therapeutic bronchoscopy. So cryostasis is a RCT, multi-center RCT, looking at spray cryotherapy for benign central airway stenosis. Reliant 2 is the next iteration of a robotic RCT, Illumicite versus Ion with integration with mobile cone beam. And then Veritas is kind of the cherry on top, at least so far, of IPOG efforts. Navigational bronchoscopy versus gold standard CT-guided needle biopsy for lung nodule diagnosis. 7 sites. We just wrapped this in just kind of the papers under review finally. This was a non-inferiority trial. Next slide here. 7 sites. So again, we don't need just the biggest and greatest and best academic centers, right? It's more difficult to generalize your results to real world application if that's all we're doing. So we had a couple sites here that were large community centers. And we need that kind of diversity in our research settings. I think is a huge plus, and certainly we'd like more of that. So this was a multi-center ICT, 7 sites. We were looking at lung nodules, true lung nodules, in the outer middle third of the lung that were adjudicated as technically amenable to both procedures by a central review committee that was only knocked out 20 of about 250 patients for being not eligible for one or the other, so relatively few. Next slide. The primary outcome here was actually diagnostic accuracy. So, but before we could adjudicate that, we have diagnostic yield, which was a secondary outcome here. Now would be probably the primary outcome we would use based on our new consensus guidance. But this was a strictly constructed diagnostic yield, and we followed for a year to make sure we did not to look for false negatives in specific benign biopsies that became malignant or were malignant. Table 1, I'm not going to belabor, but these are relatively small. I guess this is now pretty typical of most of the data coming out. We're going after smaller stuff, so 15 millimeter median nodule size. Next slide, and then just cutting straight to the chase here. We were non-inferior, so this non-inferiority trial. The rate of yield and accuracy here were very similar. Certainly these were non-inferior. Nav bronch was non-inferior to transthoracic needle biopsy with more than 98% confidence or 98% probability. Next slide here. And then complications, not surprisingly, were significantly greater in the transthoracic needle biopsy group. Particularly of interest is this needing admission or chest tube, which was 11.5% in needle biopsy and less than 1% in nav bronch. So we conclude from this next slide that navigation bronchoscopy was non-inferior to transthoracic needle biopsy and led to far fewer complications, and therefore probably should be the procedure of choice for lung nodules amenable to both procedures. And obviously, if it's not amenable to transthoracic needle biopsy, then it should be considered bronchoscopy. Important trial, and I think shows the power of what a big collaborative group can do. And these are the kinds of trials we hope to incubate and get out there and get kind of more robust data on what we're doing.
Thanks, Rob. Great. Let me ask you and maybe Jeff a quick question then. There's a really famous anecdote about Jean-François Dumont, who obviously was the father or one of the pioneers of rigid bronchoscopy, and certainly father of stents and laser in the airway and so on. When he first presented his stuff at international scientific meetings, then you get a bunch of people telling him, well, you don't have any data. All you're showing is, case series and there's no control. And so in response to that, he showed a video of somebody gasping for air and about to die. And then the video after the rigid bronch of the patient walking out of the hospital doing just fine and probably going to smoke another cigarette. But I think it's illustrative of what we hear a lot, which is, well, why in the world do you need RCTs, and how in the world do you expect the results of RCTs? Were you able to do them to generalize to my patient population? And so, question for you and Jeff is why and when are RCTs important to conduct, and what do you both see as main barriers to getting these done?
So, I think RCTs are, I think, still very important, particularly in our, there's the joke about, do you need an RCT to show parachutes work, right? We've got a bunch of really fancy parachutes, and maybe some of them have some holes we don't know about because we don't really look. We're not. These are all created by industry. These devices, and I think it is incumbent on us to kick the tires as hard as we can to make sure they're working. And maybe they are equivalent. Maybe we show that there's a lot of ways to skin the cat, and I think that's fair and valuable. But also recall that the history of medicine is littered with ideas and concepts that were felt to be so self-evidently clear that it is what we should be doing until 5 or 10 years later we actually studied it and found, in fact we were actually hurting quite a few folks. I mean, you can go look at the cardiology literature or the laparoscopic surgery, early laparoscopic surgery literature, and find a dozen examples of this. So I think, and then from a systems perspective, if I'm a hospital administrator or someone holding the purse strings at a place, and you tell me you want me to buy a thing that costs twice as much as what you have now, okay, but it'd be great if we could go to our administrators and say, here's how this is the incremental benefit that we're seeing, and we've got data showing that this will benefit our patients for XYZ. Now, whether they care about that, who knows, and that's not really on us. But I think that this helped bolster our ability to get the things that we want.
Yeah, I agree with everything Rob just said. Just keeping in mind, like, this is not parachutes we're testing, right? These are not patients in distress that need a life-saving procedure. These are diagnostic tests where there is potential equivalence of different approaches and different modalities out there. I would say the number one barrier to getting these done is that the FDA is not really forcing anybody's hand to do this. RCTs happen, which are mainly large RCTs are being funded by industry because they need, and this is more on the pharma side of things, they need these trials to go with their initial application through the FDA so they can sell their product. With the 510(k) pathway that Rob really laid out there, they don't need to do that. So all they need to do is prove that it's equivalent to something that it was in the past, and they can go out to market it. So why would a company ever risk putting itself up against anything else? There is only dollars to be lost. So I think that's where it comes back to us now. And then I think this is where like IPOG has really taken a stand, and it really fills a need where we need to pump the brakes on things a little bit here. And say, hey, there is equivalence with these different technologies. We don't know which one is better. We need to put these head to head and side to side and actually run these large scale trials. And one of the biggest, you were asking what are the biggest hurdles is honestly funding. There's not a lot of government money, meaning NIH, DoD kind of grants that are going to be totally designed for studying post-market devices that are already out there and being used. So it's up to us to be creative about how we design these trials, that we can do them with minimal budgets, to tell you the truth, to actually get these done, so we can have solid data out there that shows superiority or non-superiority of any one of these approaches.
Yeah, this is great. Thank you. I think you're quite right in that the 510(k) pathway is a needed pathway. I mean, I see both sides of the argument. Companies, device companies in general, except for the very few, very rich ones, just don't have the deep pockets to run multi-center clinical trials. Without the 510(k) pathway, we don't have many of the incredible innovations that have unquestionably advanced patient care. The flip side is that it'd be nice to have data at some point, and how do you fund that? How do you generate that? Unless you get a carrot or a stick to make this happen, it's really difficult. The drug companies have the same kind of thing, right? We get some expedited pathways for promising drugs that get to market without a typical phase 3 pivotal trial, but then they're on the market contingently, provisionally, until they have generated data showing that in a well-designed, randomized trial, they actually can confirm the claims of the earlier phase trials. And I think that's where probably the field needs to go. Sure, you get a great new robot. It looks awesome, conceptually makes sense. Let's try it, but let's enroll patients, and maybe you have reimbursement for the procedure contingent upon enrolling the patient into a study. I mean, that's been done before with the NET trial, for example. So, okay, great. Well, Lonny, do you want to say a few things, and then we need to let Diana talk about the big and exciting things that are upcoming that everybody should get involved with.
Yeah, thanks. We only have a couple of minutes, so let's just go to Diana.
Hi, everyone. I'm actually here to echo Fabien that IPOC is not a secret society. I'm actually here to highlight how you can get involved in our ongoing IPOC research group efforts. So first off, there'll be our third annual IPOC conference on minimally invasive lung cancer and pleural disease diagnosis and therapeutics. It's focusing on integration of artificial intelligence and IP. It'll be held between April 23rd to the 25th at the Johns Hopkins University. We'll be reviewing actually just a preview completed trials in the past, so Veritas Frostbite 2 liquid trial, Reliant and Tactics study, and ongoing discussion regarding our studies that are coming up, MIST-4, Streamline, Rose Gold, Sequence, and Frostbite 3. And we'll also have keynote speakers to discuss civil trials and AI in robotic-assisted bronchoscopy, some of the applications in our gastroenterology as well as pathology. And we'll also be reviewing the ongoing partnership and research efforts with Alliance for Clinical Trials in Oncology that's spearheaded by Dr. Maldonado. And next, I'd like to also review our ongoing, so first, this is all going to be published in our website, ipogresearch.org. I'll have a last slide that highlight all the links. This is our first annual Cope and Wong Next Generation Scholar Travel Award for young investigators in advancing the science of IP. So this award is designed to be given to early career physicians who represent Dr. Wong's spirit of innovation in pulmonary diagnostics and advancement in therapeutics. So some of the eligibility criteria are listed, and it'll be posted on our website. Again, and the deadline is March 10th of 2025. Eligibility in brief is tenure, faculty appointment within academic affiliation, primary and leading role in a project, and demonstrated focus on innovation in bronchoscopic diagnostic or therapeutics. So there will be prizes, first prize and second. That includes complimentary meeting registration, IPOG, dues for first year, airfare and hotel accommodations, and presenting the abstract in person at IPOC meeting. I'd like to also highlight the new Young Investigator Forum, where abstracts submitted by young investigators will be presented. It'll be an excellent networking opportunities where you'll get to meet leading experts and also develop future mentorship relationships with the faculty here. So I think it'll be a really awesome opportunity. There'll also be some educational webinars through ATS Thoracic Oncology Assembly that we'll also be addressing. So I'd also like to encourage everyone to get involved in our research efforts by joining our RCT. So 2 actively enrolling studies are Frostbite 3. It's RCT, a mediastinal cryobiopsy versus TBNA for NGS. That was discussed by Jeff, as well as Sequence. It's an assessing order of peripheral nodules biopsy and lymph node evaluation. So is it better to evaluate the lymph node prior to the robotic diagnostics biopsies or vice versa? So that's a study also enrolling currently. And lastly, this is a slide with all available links in various platforms. So we have our website here, ipogresearch.org. So this week, we'll have updates regarding how you register for our conference, the contact email for IPOG, and X and LinkedIn.
Thank you, Diana. So I guess we're at the top of the hour. So I'd like to thank the American Thoracic Society for organizing this. I think this was really helpful. I want to really invite all of you to reach out to us and get involved in some fashion. You know, if you're interested in studies, you know, we've got quite a large portfolio of studies you can get involved with. This does not necessarily imply you'd be part of a randomized control trial if you don't have the infrastructure available at your institution, but certainly we can get you involved with other simpler projects. So write down on your calendars the date and time for the IPOG meeting, and we look forward to seeing you all. Lonny, you want to say 2 words?
Yeah, well, thanks, Fabien, for running this, and everybody else for giving some great talks. And we've come a long way. So I really encourage everyone to reach out who's interested in clinical research to get involved. We have a lot of opportunities, and look forward to working with more folks and moving the field ahead.
Thank you, everybody.